This is Chapter 12, Part 1, and this is on the cell cycle. So the first question is, why do cells divide? There are three reasons. There's for reproduction, growth, or for repair and renewal. And for reproduction, mitosis, the division, is only going to occur in one-celled organisms. So very simple organisms use cell division for reproduction. And in that case, they reproduce the entire organism because they're one-celled. But for anything that is larger, any multicellular organism, cell division is going to provide for the growth and development that begins with a fertilized egg and grows to a multicelled organism, or it's going to be for the repair and renewal of cells that have died from just normal wear and tear. The most important thing in cell division is that the right things and the right amount of those things, those structures, elements, get into each of the daughter cells. And what is passed on to the daughter cells is an exact copy of the genetic material, the DNA, and also all the organelles in the cytoplasm, the cell membrane, all the enzymes that are needed for that cell to have a functional life. So what happens through mitosis is that copying of genetic material, and it actually, we'll look a little bit later on here, it's actually occurring before mitosis even begins. And then during mitosis, those organelles in cytoplasm, the cell membrane, and enzymes are divided up into the different daughter cells during the process of cytokinesis. So both prokaryotes and eukaryotes have to distribute this identical genetic material between the daughter cells. So that's one thing that they share in terms of the reproductive process. The only exception in a division process is that special type of eukaryotic cell division that we call meiosis. And that is only for the production of 4 non-identical daughter cells that each have half the chromosomes of the parent. And of course, we know that those are the sperm and the eggs, and that they are only produced in the localized structures of the gonads, either the ovaries or the testes. When it comes to the replication of the DNA, so when we're getting that exact copy of genetic material, what's really remarkable is how reliable that process is, because in most cases that DNA gets passed along to the next generation without being diluted or distorted. So that's really remarkable when you think about how many cells are going through mitosis all the time. Each, as you know, eukaryotic species has a characteristic number of chromosomes that are unique to it, and they're located in a eukaryotic cell nucleus. So for humans, our somatic cells, which is everything except the sperm and the egg, have 46 chromosomes, and that's made up of 2 sets of 23. One set from each parent. But the gamete cells, the sperm and the egg, have half that number. They have 1 set of chromosomes, which is 23 chromosomes. So in terms of that growth and development, a cell's genetic information packaged as its DNA is called its genome. In prokaryotes, the genome is often just a single long DNA molecule, but in eukaryotes, the genome can consist of a number of DNA molecules, as we were just discussing. So each of us inherited 23 chromosomes from each of our parents, 1 set in an egg and 1 set in the sperm, for that total we need of 46. And then those chromosomes, those 2 sets, were combined in the nucleus of a single cell when the sperm and the egg were united to form the zygote. The zygote then starts undergoing cycles of mitosis and cytokinesis until we get a fully developed multicellular human being made up of 200 trillion somatic cells. So this is quite a process, starting from one joined egg and sperm growing into this multicellular organism that we are. And then these processes of mitosis are going to continue every day throughout the life of an organism like us to replace any dead or damaged cells. So this process of mitosis, this repairing, and even during the process of growth and development, that process is in essence producing a clone, which means a cell with identical genetic information. That replication process, that creating new DNA for each mitotic event, is basically copying about 2 meters of DNA. So if you put all of our chromosomes together, it would line up to be about 2 meters. And those 2 meters of DNA are what gets separated into the 2 copies so that each daughter cell ends up with that complete genome. The process of mitosis, in review, consists of these 6 or 6.5 basic steps. So mitosis starts with interphase, which really there's not a lot going on during interphase. But then the active part of mitosis really begins with prophase. So that we have prophase, prometaphase, metaphase, anaphase, cytokinesis, and telophase. And the telophase or telophase and cytokinesis are often grouped together because they almost happen simultaneously. It's so quickly occurring. So we're going to break this down. First, we'll look at interphase. So interphase is where a cell spends most of its time. Interphase itself is divided into 3 phases. So if we think of the cell cycle like a clock, interphase consists of 3/4 of that clock, and then mitosis is only 1/4. And the 3 phases of interphase are these ones that are here, the largest portion of the cycle. And the divisions of those of interphase includes a G1, an S, and a G2 phase. The G1 stands for first gap or first growth, and it's really when the cell is just doing its everyday job. There's some cell growth going on during this period. And then the S phase is when we have synthesis of DNA occurring. So this is actually when the DNA is being copied or replicated. Then we have a G2 or second gap growth phase. And this is when the cell is actually getting ready for division. This is when it's duplicating its organelles, making copies of proteins, making more membrane material, and there's a little bit more growth going on. And then if the cell's going to start dividing, it will go right into mitosis, which is broken into those major phases we just talked about. So first, we'll look at the first phase of interphase, which is G1. And this is where 90% of a cell is going to spend its time. Again, a cell's everyday job varies depending on where it's at. But for example, part of its everyday job could be producing RNA or synthesizing proteins, making enzymes. Basically, it's just doing the life of a cell. It's the work phase. This will continue until there is some trigger that sends G1 into the S phase, which means we have the duplication or synthesis of the DNA ready to occur. During this phase, also, I want to mention that the DNA is loosely packed in long chromatin fibers. So this is an example of how a cell can actually, depending on what type of cell it is, spend different amounts of time in that G1 phase. So cells are going to go through mitosis, and at the end of mitosis, they go into the G1 or GAP1 phase. And most cells are going to grow and mature and divide again. So they're going to continue to go through this G1, S, G2, and M process, this cycle here. So things like epithelial cells, stem cells, blood cells would be all types of cells that would just continue this cycle around. But there are also cells that really don't continue to grow or to go through mitosis and divide. So these cells just grow and mature, and they go into what's called a G0 or a resting phase. And they never return back to the whole division process of the cycle, going through synthesis, G2, and mitosis. So examples of that are brain or nerve cells or muscle cells. Also, liver cells can go into G0 phase, that resting phase, but they are also able to come out of it. So they're an example of a type of cell that can go between this resting phase and the active phase. Some cells in a multicellular organism are in between these two things. They do divide, but they don't do it frequently. In terms of timing, a typical human cell might divide once every 24 hours. And of that 24 hours, the mitosis phase is actually not going to last more than an hour. The S phase might take 10 to 12 hours. So that duplication process, replication actually, is about half the cycle. And then the rest of the time is going to be divided between G1 and G2. But again, that G1 phase is the one that varies the most in length from cell type to cell type. So just a review of DNA itself. DNA molecules are packaged into chromosomes. We have that double helix DNA molecule that is actually wrapped around proteins that are called histones. And together, that complex of protein and DNA is called chromatin. And that's that word right here, that protein DNA complex. The DNA itself can carry several hundred to a few thousand genes. And those proteins, the job of the histones is to maintain the structure of the chromosome. And we'll learn when we move on into genetics that histones also help control the activity of genes. So after G1, after that growth phase, if a cell has a signal that it should continue on to mitosis, it will go into S phase. And that's again where we have copying and replicating of DNA. So when a cell's not dividing, each chromosome stays in the form of that long, thin chromatin fiber. But for a cell to actually replicate, the DNA has to be separated so we can actually have a correct copy made. And again, that remarkable feature of mitosis in cells is that the error rate of replicating DNA is 1 per 100 million bases. So it's a very small error rate. And there actually is a mechanism, again, when we go into genetics and study microbiology, we're going to look a little bit more closely at that. But if we think about our DNA like a